Today we will study about polyembryony. Maheshwari in 1950 defined polyembryony as the occurrence of more than one embryo in a seed. Since the additional embryo generally degenerate in the course of seed development, the development of more than one embryo is frequently not seen. Polyembryony has been found as a regular feature in citrus and Magnifera. Types of polyembryony. Polyembryony was first reported by Leeuwenhoek in 1719 in certain orange citrus seed. Later, a considerable study was made by several embryologists like Braun 1859, Ernest 1918, Weber 1940, and Sneffer in 1929 divided polyembryony into two groups: the true polyembryony, false polyembryony. In true polyembryony, in which there is a development of multiple embryo within or by projection into a single embryo sac. False polyembryony, in which the multiple embryos are developed from different embryo sacs. Gustafson in 1946 proposed that the true polyembryony is the development of embryo derived from one or several embryo sacs present in the same nucellus. The term false polyembryony should be restricted to those cases in which two or more nuclei, each with its embryo sac fused, and the seed may appear single or double depending upon the degree of fusion. Born 1959 classified true polyembryony into four types: cleavage of zygote or zygotic embryo, formation of embryos from the cell of the embryo sac other than the egg, development of more than one embryo sac within the same ovule, activation of some initial sporophytic cell of the ovule. There are there are following types of the polyembryony. First is the cleavage polyembryony, where polyembryony arise by the cleavage of the zygote or the proembryo into two or more units. The condition is called the cleavage polyembryony. Cleavage polyembryony is less common in angiosperm, while it is commonly found in the gymnosperm. Among the angiosperms, it is a very common in orchid. Rao 1965 reported that the apical meristem, promeristem of the embryo in Vanda or divides it into a number of primordial cells 3 to 9, each further organizing into an embryo. Swami 1943 reported it in the Eulophia epidendria arising in the three different ways. Zygote divide irregularly to form a mass of cell, of which those lying toward the chalazal end grow simultaneously and give rise to multiple embryo. The proembryo gives out small bud or outgrowth which may themselves function as an embryo. The filamentous embryo become branched and each of the branch grow into an embryo. Jeffrey in 1895 reported the cleavage polyembryony in Erythronium americanum, Ernest 1910 in Tulipa gesneria, Hall in 1902 in Limnocaris emarginata, in Coccus nucifera, Rao 1965 in 19. Abnormal type of cleavage polyembryony has been reported in some cases. Cooper 1943 reported in Nicotiana rustica an ovule with two embryo, of which the smaller had apparently arisen as an outgrowth from the apex of the primary embryo. Abnormal embryo arising from the suspensor has been reported by Crete in Lobella syphilitica by Subramaniam in 1946. Embryos from the cell of the embryo sac other than the egg. Such type of embryo may arise either from the synergid or from the antipodal. Embryo from the synergid. Embryo from the synergid are the most common source. Some embryos may be haploid or diploid depending upon the development from unfertilized or fertilized synergids. Synergid may be fertilized either by the entry of more than one pollen tube into the embryo sac or by the presence of additional sperm in the same pollen tube. Embryo arises from the fertilized synergid have been reported in Nazar minor, Telema grandifolia, Fragaria vesica, peltifilum peltatum, Aristolochia brachyta, Poa alpina, while from the unfertilized synergid in Argemon mexicana, Plantago lanceolata, Fasciolus vulgaris, and Lolinium species. Embryo from the antipodal cell. Formation of embryo from the antipodal cell is little known. These have been reported in Phaspalam uh, scrabiculatum. Embryos arising from the endosperm cell. The formation of embryo from the endosperm cell was reported by Truebert in 1828 in the Balanophora elongata. Also, in Kazeler 1942 also reported additional embryo from the endospermal cell. False polyembryony, more than one embryo sac in an ovule. The term referred to the embryo arising from other embryo sac found within the same ovule. Such embryo sac in an ovule may arise from the derivative of the same megaspore mother cell, derivative of two or more megaspore mother cell, or from the cell of the nucellus. Kumar and Joshi 1942 reported that the polyembryonic condition may also arise by the fusion of two or more ovules in case of Rhizophora mucronata. The multiple embryo sac in an ovule have been reported by Citrus, Poa pratensis, and Crassudia equisifolius by Swami in 1948. Twin embryo in a seed have been reported in many cases, and these may be formed in a different combination, diploid in a geamage, or diploid triploid in Triticum vulgaris, and in caecale, or haploid triploid in phylum, or haploid diploid in geamage. Thank you.